to our presentation, seed viability of Lonicera macai, also known as bush honeysuckle or honor honeysuckle, in the seed bank after parental plant removal. This research was done by me, Brielle Lunton,
Kara McGraw,
Carly Astalos,
Michael Williams.
Alright, so we're all kind of brain dead by this point in the semester, so I'm not going to talk about a bunch of complicated science stuff. I'm going to talk about aliens. Now we all know aliens are real, right? I mean, they gave us mathematics and the pyramids. We couldn't have done that ourselves. But the government just keeps trying to cover it up. It's really weird, but they just keep trying to cover it up. But the aliens keep coming back. This is analogous to honeysuckle, which is an invasive species. Which means that it's not native to the ecosystem here in America, at least this particular species of honeysuckle. Invasive species can kill off native plants. They outcompete them for resources and nutrients such as like light and pollinators. And also they're typically not subject to native herbivory, so they're not being preyed upon by herbivores, but they also may benefit from their berries being eaten by birds and deer and then being dispersed. Now, specifically, we wanted to investigate the presence of honeysuckle within the seed bank. And the seed bank is basically just the soil where plants have deposited their seeds, which may germinate in subsequent years.
Honeysuckle is in the Lonicera genus. It is native to Northeast Asia and Japan. Actually, in Japan right now, it is considered an endangered species. It can also be found in North America and Europe, though, of course, here it's considered an invasive species. Honeysuckle can grow up to 16 feet tall and produces thick green leaves sooner than native plants. It also produces an allopathic chemical that poisons the other plants around it and causes them to germinate later. There are 3 types of removal for honeysuckle: uprooting, cutting, and burning, though none of these affect the seed bank. In our experiment, the honeysuckle was removed through uprooting. We wanted to know how long honeysuckle remains viable in the seed bank. We hypothesized that after 5 years since removal, there would be less honeysuckle germinates in the seed bank than 2 years since removal, and 2 years since removal would have less honeysuckle seed germinates than where honeysuckle is still present. We took soil samples from Martha Liffey Thompson Nature Sanctuary. The soil samples were from 3 10 by 10 meters wide plots, one that still had honeysuckle present, one that had honeysuckle removed 2 years ago, and one that had honeysuckle removed 3 years ago. 5 years ago.
Okay, so for our experimental design, one of the main things we had was a map from the director of the Nature Sanctuary, Michael Sandy, that had information about where the honeysuckle was removed was at. And so we used a soil core that had a 1-inch diameter, and we marked it at 10 centimeters with a Sharpie to properly breach the seed bank when we collected the samples. We had 15 samples per plot, and each sample was placed in its own separate bag and marked with its TSR, which is the time since removal. Oh wait, go back. We also collected about 100 berries and placed them in a bag on their own to use them as a control group and plant them when we got back to the greenhouse to examine honeysuckle germinates alone. And so when we got back to the greenhouse, we had 60 pots. They're like little green pots of a 9-inch diameter. We put paper towels on the bottom and we filled them 3/4 of the way full with sand. And the paper towel was used to keep the sand from running out. And then along the top, we put the samples on and we marked each of the pots with the TSR, and we gave them a number from 1 to 45. And then the last 15 pots were used for the berries, and they were sifted for seeds and emptied in the last 15 pots and labeled berries with numbers 46 to 60. So like this one has time since removal is 5 years, and it's pot number 45. We watered the plants every day, and we always did a data collection. We shuffled the pots around every 2 weeks to make sure that none of the samples from the same area were by each other. And every time we saw germinates, we would use these little colorful toothpicks to mark where we saw them. And on our data sheet, we would mark how many we saw and what plant we saw them in.
We ran a total of 3 tests performed through our studio: the Kruskal-Wallis test, the Levine test, and the Shapiro-Wilks test. The Kruskal-Wallis test shows significant statistics when compared to the Shapiro and the Levine test. The Kruskal-Wallis test was significant with a p-value of 0.08. The Shapiro and the Levine tests were used to determine the best way to propose our data. They confirmed if we could use an ANOVA. ANOVA was typical for results like we had because it was compared among groups, though they were not useful because we had so many zeros within our data. Through the Kruskal-Wallis test, we made this graph, which is the total median germinates per pot per treatment. The x-axis is the time since honeysuckle removal for the 0-year, 2-year, and 5-year. The y-axis is the number of germinates per pot. The graph compared the medians of our results. The medians are the thick black lines. The dot over the 0-year is just a statistical outlier that we had. The graph is labeled A, B, and B to show the difference between our results. A and AB and B and AB were not significant to each other. The A and B were significantly different to each other, meaning that that's where we saw an increase in our germinates.
So for our discussion, our hypothesis was neither supported nor it was proven false due to us having no honeysuckle germinates at all. We did have a total of 28 germinates, but none of them proved to be honeysuckle. This could have been due to the lack of honeysuckle seedlings in the soil samples because we did take fairly small samples of the soil. It could have just been we didn't get any seeds in the samples. It also could have been because the conditions were not optimal for honeysuckle germination. Like there might have not have been enough nutrients in the soil that we collected, as well as native plant seed replenishment. So other animals, like animal droppings, replenishing native plants in the seed bank could have been the reason why we had so many germinates that were not honeysuckle. And for our future study, we could actually focus on seed viability. In this study, we didn't focus on seed viability due to not having any honeysuckle germinates. We can also focus on the impacts that the honeysuckle has within the soil, like Carly mentioned, the allelopathic chemicals. Also how honeysuckle enters ecosystems and establishes itself, and then the conservation, restoration of native plants by reducing the seeds in the seed bank of honeysuckle, which can be done by consistently removing the honeysuckle annually. So for our plots we collected was 5 years removed, but 5 years later it wasn't removed again. So it wasn't consistently removed, which could have, led to the honeysuckle just replenishing itself in the seed bank. So consistently removing them would help reduce the amount of honeysuckle seeds in the seed bank. And we would like to thank Professor Nickel and Dr. Schaefer for helping us with our experiment and acting as mentors.
Thanks to Dr. David Gorchov at Miami University for giving us information about honeysuckle germination and seedlings.
To Williamsville College for the use of their greenhouse and materials.
And to the Martha Luther Thompson Nature Sanctuary and the director Michael Sandy for providing us with information on the honeysuckle plots and also letting us collect the samples.
Literature cited page. Any questions?
Vigorous applause. I'll stop clapping when Muhammad claps too. Thank you. Questions? What are those aliens from?
I think they're from Doctor Who.
Oh, okay. That's one I haven't watched.
Yeah.
Oh.
Yeah.
So you said you didn't grow any honeysuckle?
No, we didn't see
We
any.
didn't. But we did continue to water them after we finished our experiment to see if we had any. And the seeds actually, the control group from the berries, they did germinate, but we still didn't have any from the actual seed bank.
Is there did you find in your research any evidence of intraspecific allelopathy in honeysuckle?
Personally, no.
Yeah.
Cool.
That's an interesting idea though. Yeah. Autoallelopathy. You mentioned in response to Christian that you use the berries as a control group. Is that really what they were used for?
Well, they were used so we know what like honeysuckle germinates look like, so we knew what to look for and when our seed bank actually germinated. So not necessarily a control group, but as like a reference.
Okay. More questions from the
Questions?
Did you guys identify any other species you did find?
No,
We did
no.
not.
That could be another potential future study.
Mm-hmm.
How big are they right now?
It varies probably from a centimeter for some of the germinants to an inch or two for others.
Yeah. So they're still pretty small. Fairly difficult to identify. We're going to keep them going so we can do some identification.
Yeah. But if you want
In the future.
we're gonna identify a Christian, yeah, you're more welcome to help us.
I think he was volunteering.
I think so too.
Any other questions?
Yes. No. So.
Front row. We need a question from the front row.
Really good on any question. Alright, well, they just have to get questions from us. How deep did you collect soil samples?
10 centimeters.
10 meters? Okay. I didn't know if you said that. Okay. So, what would be the potential effect of uprooting on the soil? So you mentioned that the honeysuckle was removed by uprooting honeysuckle. So what would be the potential effect of uprooting species on the soil seed bank? What happens when plants are uprooted?
It's harder for them to grow back, but I don't know of any direct effect that it would have on the seed bank.
Well, maybe I don't understand how uprooting happens, but what happens when you uproot a plant? What are you doing?
Pulling it out.
Okay, you're pulling it out of the ground. So what happens?
Are
when
these
you-
plants large
what
or
happens
are
when you
these plants
pull a plant out of
small?
the ground?
Oh, but it could take some of the seeds with it, you're saying. If you uproot a plant, you could probably take some of the seeds from the plant out of the seed bank by removing the soil.
If you're taking soil with it, but what is happening to the soil that stays behind?
It's being disturbed.
Sure, it's being disturbed. So I guess what I'm wondering is, do you think that uprooting as a mechanism for removal of honeysuckle might have a different effect on what you might find compared to if honeysuckle was just cut and sprayed?
Well, I was reading that honeysuckle actually does better in disturbed plant communities, so that could potentially help it germinate more.
But like I also read that birding also helps honeysuckle like germinate more, so I guess it doesn't really any kind of disturbance. You know, they would still grow even if you uprooted it and it disturbed the soil. It would still grow just as much if you like burn it.
Okay. The other thing I'm wondering potentially too is if it brings up soil from deeper down and deeper soil gets placed on top of higher soil, it's going to potentially influence seed burial depth, and so that could potentially have a different effect than if you didn't disturb the soil anyway in terms of what you might find.
Kind of related with the soil, thinking about the soil a little bit, you mentioned allelopathic effects. And I know you probably don't know, I wouldn't expect you to go into depth in understanding how that works, but I was wondering if you might know just in general what allelopathic effects are on other plants.
Honeysuckle, there was a study done, honeysuckle can delay germination, not necessarily consistently across all other species of plants, but certain species are more sensitive to these chemicals and can delay their germination.
Okay. So allelopathy mostly affects seeds that are in the.
At least that's what I was
Right.
reading, yeah.
Okay. By
mentioned that the honeysuckle was endangered in Japan, but it's an invasive species here. You guys know why that is? Is the allelopathy not as effective with species that are native to Japan? What's the reason for that?
So we're not exactly sure why it's endangered in Japan, but we know it was introduced here as a combatant against erosion as far back as like the 1890s, I want to say. But so one of the dispersal methods is like the American robin eats the berries and its seeds are spread around, and so that could have contributed to it becoming invasive because it has the qualities to grow in various environments. So it's like it can be in partial shade or like full sunlight, and it can grow in wet and dry climates and areas, so to speak.
In some cases, allelopathy might be more effective in invaded ranges because the native plants in that area like aren't used to the chemical, whereas like plants in the native range have had more time to be exposed to it and potentially have evolved to be less susceptible in some way. So it could partly be just the novelty of the chemical making it have a greater impact in an invaded community. Why did you guys shuffle the pots around in the greenhouse?
So they all got like the same treatment. So it wasn't like one was getting
They
more
all got the same what?
same treatment or like same.
What do you mean
I guess
by
not
that?
treatment, like
Temperature and sunlight.
yeah, like the same environment, I guess. So that one wasn't like had no sunlight and like the other side didn't, or like there was like more wind and like on one, you know. So they all were like they had the same environment, I guess is a better way to say it.
So to control for any variability in environmental
Yeah.
conditions in the greenhouse. Okay.
Why did you use sand in the lower part of the pots?
So that the soil samples that we did collect, none of the seeds in the seed bank would be on the bottom, so it wouldn't be able to germinate.
Why sand though?
Because it doesn't hold too much moisture and like water is leaving through it. So like
Why would you use sand rather than collect soil from behind the greenhouse?
So as not to have like other seeds in it, like so it doesn't germinate like other plants. I guess I guess that makes sense. I don't know how to
What
word
about
it.
potting soil though? Potting soil doesn't generally have seeds in it. So why wouldn't you just put some potting soil on the bottom and then your soil on top of that?
So that the seeds wouldn't be exposed to a different variable, I suppose.
Well, they're all exposed to the same variable. If all the pots have soil, potting soil in the bottom, they would all be exposed in the same way.
I don't know if they like know why we did it. So it's answering.
That's okay.
Can you go to your graph, please? Alright, so you found that you said you didn't find any honeysuckle germinants, but you found more germinants in the 5 years since honeysuckle removal than where there's still honeysuckle present. So you gave reasons for why you might not have found honeysuckle, but what are some reasons why you think there are more germinants in the sites where honeysuckle has been removed, or was removed 5 years ago?
Native plants may have had a chance to replenish the seed bank, and that accounts for the higher germination of native plants that we saw.
And it could also be due to the 0 year stands as it stands for like where the honeysuckle is still present, and so when honeysuckle fully grows, it has very dense coverage due to its leaves, and so it could really just be keeping other plants from germinating by blocking out like sunlight and taking up the nutrients and stuff.
So do you think the seeds that germinated in the site that's 5 years since removal were in the soil seed bank at the time honeysuckle was removed? Or do you think that those are seeds that have dispersed into the site from other areas after honeysuckle was removed? Or had come from plants that had just were growing
Probably a
after
mixture
the
of
removal?
the last two, that it came from other areas around that after the honeysuckle was removed, germinants had grown and produced seeds in the area.
Can you describe the 3 plots a little bit in terms of what you saw with vegetation currently?
Well, I know that the 5 years since removal was, since it's been removed so far, there was like a lot of honeysuckle, but there's also a lot of other like tall grasses and stuff that were there as well. It seemed like there was a lot more vegetation than in the other plots, a lot more native vegetation.
So you're saying that there's some grasses and some other vegetation, but you said there's also a lot of honeysuckle. Can you?
Like,
sort of
I guess it wasn't
not
technically a lot of honeysuckle.
I don't know.
There's like a few bushes. The bushes that were in the 5-year plots were pretty tall, pretty big, so I guess there wasn't like a lot per se. There was just bigger.
What's tall and big? Use your arms.
Like, some of them were really tall, like as tall as us, but the plots.
In the 5-year plot?
Was it in the 5-year plot? Yeah, I think the 5-year plot they were pretty tall. Yeah, all of them were pretty tall in the 5-year plot.
But then there was still grass and stuff growing there?
Yeah, well, because there was only a few bushes, so it wasn't like a lot of honeysuckle. There was just like tall, if I remember it right. Am I like, yeah, like it was on the trail, and there was like, they were pretty tall and like, I guess like wide, but there was only like a few, like 2 or 3 bushes with other like grasses and stuff around. If I'm remembering it right.
Okay. So were there any young honeysuckle bushes growing?
In the 5-year plot?
Yeah.
Not that I can remember, unless anybody else can. I don't really remember any young. They're all pretty mature.
What did the 2-year plot look like?
I think there's actually a picture like one of these if you like go back. That one. I think which one is this is one of them. I'm trying to think which one was it though.
No, I took this picture.
Oh.
Yeah, this one.
Okay.
Just kidding.
Just kidding. But no, at the 2-year plot, wasn't that the one, I feel like that was drawn on like a slope. So like it was a slope upwards, and so like there was a lot of honeysuckle like around the edge, like close to the edge of the walkway. And like that's where we saw like a lot of the berries growing on it, and then like it got like it moved up the slope, but probably if you like line it up, it was all about the same height. It wasn't like 16 meters or feet or anything like that, but it was fairly tall as well too. I want to say personally for me, it probably came like right here to me.
Okay.
And then in the 0-year plots, it was just everywhere we turned.
And in the 0 year, they were pretty, I guess they were baby plants. They weren't as mature as all the other ones. In the 0 year plots, there were smaller bushes, but it was everywhere.
Okay, so there was a lot of small honeysuckle in that one.
Yeah, in the 0
Kind of
year.
throughout.
So we're talking about the 2 to 3
How long can seeds stay in the seed bank on average? Do you see like a limit? I don't know. I was just curious.
There's not really an average. It varies a lot. Like you probably have to look that up.
So before honeysuckle occupied that area, it's possible that the plants previously there remained in the seed bank and grew back after it was removed.
Yes.
Years.
So you were saying that there's honeysuckle in the 2 and 5 years since removal sites, right? Do you think those honeysuckle came back from grew from seeds, or from some resprouting from not removing all of the below ground structures?
Well, personally, I think they like for the 5 years, would have just been seeds from the seed bank that have grown out because we didn't have any actual honeysuckle germinates in the 5-year samples. So I think that the 5 years was just, it wasn't redispersed from animal and stuff. The 2 years very likely could have been because we didn't have as many germinates, but I think that the 5 year was just had grown out of the seeds from the seed bank.
Do honeysuckle seeds require any special treatment or conditions for germination to occur?
They need to go through cold stratification, which I can't remember specifically.
I think the beginning is I want to say, I got it in my paper in the lab report, but I can't remember it off the top of my head.
I know they need to do for a period of 6 weeks. I can't remember the
exact temperature, and then after that I know in one of the studies that Dr. Gortop did, where they studied the white-tailed deer and the dispersal of the honeysuckle seeds, they went through the cold stratification. I can't remember the first one, but after the 6 weeks they were in 15 degrees Celsius during the day, and I want to say 24 degrees Celsius at night, or it's switched around or something like that.
Why do you think it's important to know how long honeysuckle seeds are viable in a seed bank?
Because once honeysuckle is removed, after so many years, that seeds are no longer viable